Hi, I'm gonna show you a virtual brain cutting of the brain, of the human brain using the Human Brain Project. I've already shown you the link earlier. Okay, so here we have in this window, this box, the brain as it looks like from the outside. You see here the different planes. This is the coronal plane. This is the sagittal plane, and this is the axial plane, this horizontal x-axis here. Okay, and this is a view of the brain. So imagine this as a cleaver, and this is what we're going to cut. So we're going to drag this brain past this cleaver to chop it into pieces on the coronal plane. So I'm going to drag it here. So this here is the frontal pole of the brain. We're going to start cutting through it. So focus your attention on this box over here. So that's the frontal pole being cut. And then you get to this point. So mostly gray matter, and then you see the white matter starts to appear. So if you put the mouse here, it labels it for you. It's the white matter. If you put it here, it's in the gray matter. Alright, so we move the brain further and keep cutting it until this point. Okay, here you see at the base of the brain the orbital gyri. This one in the most midline is called the straight gyrus or straight gyri. So we'll keep dragging it now. Move it some more. Did we reach this point? This is what I call the triceratops view of the frontal lobe. So imagine these are two triceratops locking horns. Okay, the horn is actually the corpus callosum which joins them. Or imagine there's a triceratops looking at itself in a mirror. The eyes of the triceratops are cavities which contain cerebrospinal fluid. This is the so-called frontal horn as it appears here, as it's being cut here. So this is a cavity, it's an empty space. In life, it contains fluid. Okay, so as you see, as we keep slicing here, you see we're approaching a mass of gray matter down here. Okay, so these are the basal ganglia collectively. Okay, and the white matter from up here inserts itself between these two gray matter masses over here, which we'll be naming in a while. So as we proceed, okay, so I mentioned that is the corpus callosum. As we proceed here, now those gray matter, we are starting to cut. So if this is the triangular shaped lateral ventricles frontal horn, okay, here on its lateral aspect is that gray matter mass, collectively called the corpus striatum. We'll see why it's called the striped body later on, but as this cut, it's not yet striped, it's purely gray matter. Alright, so this gray matter, which forms the lateral wall of this frontal horn, is known as the caudate nucleus. So we're cutting through the caudate nucleus, and we're going to move some more until we see here it's now actually split into two. So this white matter structure here is called the internal capsule. It's anterior limb. So we'll get into that later. What is an anterior limb compared to a posterior limb of the internal capsule? So the caudate nucleus over here has been separated from its counterpart here, which is known as the putamen. And the part where they still intersect here is called the nucleus accumbens. Now I'm gonna go into this into further detail in the limbic system. Just know that this structure is what motivates you because it's part of the reward circuits of the brain, including this structure over here. So coming down here from the septum pellucidum, or the separator of the two lateral ventricle chambers, the septum pellucidum, there are two slivers of gray matter inside. These are called the septal nuclei. So together, the septal nuclei and the nucleus accumbens make use of your dopamine neurotransmitter to reward it's what makes you feel an experience is rewarding or not, thereby having an effect on your motivational levels. So if you don't find any motivation in what you're doing, it's because you don't see any point. You don't feel any reward in pursuing it. All right? So we'll proceed. As we cut further, we bring the brain forward. Let's just drag it down here. So you see here the separation now between the caudate nucleus and the basal ganglia. So far it's known as the putamen. You still see here the septum, the septal nuclei here. So this is very hard to see on the gross specimen because they're very small. So at least with the computer modeling that we have, we can actually zoom in and fix the layout here on our screen. Okay, so caudate nucleus, the internal capsule, its anterior limb, and the putamen over here. Then we'll move the brain some more until it is fully, the two gray matter structures are fully separated from each other. Okay, at this point, we are now reaching the frontal lobe over here. We have started to cut the temporal pole of the temporal lobe over here. Because our marker for this being the temporal lobe, aside from this Sylvian fissure here, separating the frontal lobe from the temporal lobe, we now have this white matter tract which connects the temporal lobe on this side with the temporal lobe on this side. This white matter structure is now called the anterior commissure. So a commissure connects right and left hemispheres. Beneath that anterior commissure is an important gray matter structure. So we don't see it that well on gross specimens because you really need very thin slices of the brain or you'll miss it. So this gray matter structure is what keeps your attention span focused, helps you learn new things and remember them. This is the nucleus at the base of the brain, formerly known as the substantia innominata, or the substance that is unnamed, into something that is named. So this is the basal nucleus of Maynard. The full name is the basal optic nucleus of Maynard because of this structure over here, which is actually the two optic nerves as they appear in the optic chiasm. Alright, so right now, just to remember where we are in the frontal lobe and the temporal poles are starting to appear. And we are right here in the middle of the corpus callosum, okay, in the so-called body of the corpus callosum. What we were seeing just now was the knee of the corpus callosum, the genu. So here, the word genuflection, so a knee. Alright, so we'll move on, bring the brain forward to this point where a gray matter nucleus appears here in the temporal lobe. So this gray matter structure here is your amygdala. It is your emotional brain, the part of your brain that feels fear, that feels anger, and other intense feelings, which is congruent with the function of this putamen over here, which is also a modulator of intensity of whatever it is you're doing, whatever it is you're feeling. So this brain within a brain here, which contains the basal ganglia and is closely associated with the amygdala over here, is the insula, the Isle of Ryle. Alright? Okay, moving forward. So we go beyond the anterior commissure once it disappears. So at this point, we see the putamen, its globus pallidus partner over here. And we see here the thalamus appearing, which forms the lateral walls of the third ventricle. So here, the lateral ventricles will communicate with the third ventricle down here using these openings, which are called the foramina of Monroe. So these openings lead to the third ventricle, and the third ventricle will lead further down into the brainstem later on. So just to reorient, we are now over here, almost close to the middle of the brain. We have sliced through most of the basal ganglia, and now we're starting to proceed posteriorly. So our marker now here, we are in the temporal lobe, but we're still anterior because we see the amygdala over here. So let's move the brain some more. You move it some more like this. Still see here the amygdala in all its glory. You still see here the thalamus with its third ventricle. You see here the internal capsule proceeding to what will become the brainstem. These two structures here are the beginnings of the cerebral peduncles of the midbrain. So that's already part of the brainstem. Here is the thalamus when it looks most heart-shaped. So you'll get to know the different thalamic parts when Dr. Baticulon discusses the diencephalon. You see here, the caudate nucleus shrinks down because this is now its tail as we proceed posteriorly. Okay, so we'll move the brain some more forward, where the amygdala starts to give way to another structure right behind it. So we'll move it further until it becomes much clearer. This is now the temporal lobe at the level where it forms the so-called hippocampus, something you've been using your entire school life. So this contains short-term memory. The goal is to get beyond short-term memory and lead to understanding. Alright, so this is the midbrain already. The structure that tells us this is the midbrain is this dark gray matter stripe here. That is your substantia nigra. Okay, so to orient, we are now here in almost exactly the middle cut of the brain, coronally. We are at the middle and smallest point of the corpus callosum. We have the appearance of the hippocampus, the hippocampi on both sides. And you have the thalamus here in the middle, forming the walls of the third ventricle. Okay, so the basal ganglia give way to this larger posterior limb of the internal capsule, which is actually continuous with the cerebral peduncles over here of the midbrain. So let's move backwards. Go through the brain. Still see here the Amunts horn of the hippocampus. Now here, starting to appear these two orbs within the midbrain. These are the red nuclei. Okay, so I will discuss later on how this red nucleus, the substantia nigra, and the brainstem all interact to form or to modify our movements. So this patient, the donor of this brain actually had a stroke. So you have this this structure here which does not belong there. You don't see it here on the other side. So it's something that is abnormal, most likely a stroke. So it forms a cavity here full of astrocytes. All right. So focusing now on the hippocampus as we move it backwards. It actually forms a tract. So you'll see the fornix here in the temporal lobe, and you also see it here underneath the corpus callosum. So how did it get from here to there without cutting through the brain? It means it went somewhere. We didn't see it anteriorly over here. So to get from down here to up there, it means it had to loop posteriorly and follow the corpus callosum. So fornix, okay, red nucleus, substantia nigra. So the fornix is the white matter tract of this primitive gray matter here, the hippocampus. So we'll move to the posterior parts of the brain, follow that fornix. So now you see here the brainstem. This is the midbrain. That is the pons, which is the fattest part of the brainstem. Okay, so most of the terminology that we use are all artificial. It's still one brain. Here, what defines diencephalon from the brainstem when they're all continuous? So it's just an anatomical, artificial term. They're all connected. So here, the corpus callosum, which is thinned out in the middle, now fattens out again. It becomes broader again as we move posteriorly. So that's the hippocampus. It's starting to disappear. Its fornices are now mostly seen up here beneath the corpus callosum. So this broadest part of the corpus callosum over here, the opposite of the gene over here, is called the splenium. So that's the splenium. So the broadest part of the corpus callosum, telling you there must be something important going on here in this parietal cortex. So there's a need to communicate something here to the other side, or vice versa, because only one side deals with language, the other does not. So the illiterate side depends on the other side for understanding. So more on that as we go through the functional parts of the cerebral cortex in phase 2. Alright, so here is the smallest chamber. So these are the lateral ventricles. The third ventricle disappeared because it now shrank into this aqueduct of Sylvius, which is found inside the midbrain. Okay, so those are the fornices of the hippocampus, splenium. Then as we proceed posteriorly, and posteriorly, there you see the hippocampus and its fornix coming in from behind, going into the area beneath the corpus callosum and then proceeding anteriorly. We will trace that fornix backwards once we reach the posterior part of the brain. Right? So that's a hippocampus, that's its fornix going to the midline. Now this is actually the exact midline of the brain. Our structure here is called the pineal gland. So I will lecture to you that in endocrinology. Okay, so this forms a very calcific part of the brain. I have calcium deposits here. It's right there in the middle, in the midline of the brain. It serves as a marker for us that this is the center of the brain. Okay, we move beyond it. The pineal gland starts to disappear, which tells us we're reaching the most posterior part of the brain over here. We don't really see the hippocampus already starts to fade from view, and we see only the fornices up here. We see here the back portion of the pons, which is part of the midbrain. We'll see later an important part of the CSF pathways once we get past this point. Okay, so here, in contrast to the frontal horns we saw earlier, these are the more pyramidal shape or the trigone of the lateral ventricle, which tells us already near the back of the brain over here. So if you're lost here, you go back to this picture. We have crossed most of the brain already. We don't see any more corpus callosum here. Now you see two separate parietal lobes over here with its counterpart of the lateral ventricle, the trigones. And we move backwards. Then we see this cavity here. So this cavity at the back of the brainstem of the pons, and that's the cerebellum on top of it. See, this is the last chamber of the CSF pathway. This is the fourth ventricle. The cerebellum forms its roof. The pons forms its floor. Up here, the trigones are disappearing. And we're getting into the so-called occipital lobe. So this is parietal up here. Down here is occipital. So this is the occipital pole of the brain. Down here, that's the occipital lobe of the brain. We are now reaching that point known as the calcarine fissure or calcarine sulcus. Okay, so we've reached the back portion of the brain. I've been mentioning in the histology the line of Generi. So if I didn't mention it earlier, this whole slice is actually a histologic section. You can actually zoom in on this calcarine fissure because the gray matter in this area is actually your visual cortex. So vision is a highly specialized sense. So it must have a very thick layer 4 on histologic section. And knowing that this is a histologic section, let us zoom in to that calcarine sulcus. Zoom in, zoom in, zoom in, and zoom in, and zoom in, and zoom in some more. Now, this is a very thick granular layer. Okay, and then because of those very thick layer 4s, that area in between forms a striped appearance, as you can see here also. So that striped appearance is seen best on histologic sections or with a very good magnifying glass for the gross specimens, if you're looking at a gross specimen. We'll see how far we can zoom in. Okay, the reason for the striped appearance is because those gray matter will have their very thickly myelinated axons. So a lot of oligodendrocytes will appear here, which will give it that striped appearance, so black, white, and black, because of the pyramidal layer underneath. So to see it again from a distance, the stripe, there you have it, gray, white, gray. So we zoom out and proceed to see what the brain looks like from the back. So that is occipital lobe because of the calcarine sulcus or calcarine fissure. Okay, all the way to the end of the occipital pulse, then moving backward again. And then those are your calcarine fissures. If you look at this box, that's the parietal occipital fissure that separates the parietal lobe from most of the occipital lobe. Let me move forward and forward again. Okay, fourth ventricle. Those are the occipital horns which appear just before the calcarine fissure appears. So those are the occipital horns. Then that's your trigone of the lateral ventricles. That's the splenium. So the splenium is your marker for the trigones. When you get past the splenium, those are the occipital horns. So that's the difference. Splenium, trigone. No splenium, occipital horns. So moving forward, hippocampus, a bit of hippocampus and its fornix over here. The splenium of the corpus callosum, the, the pineal gland or pineal gland here in the middle. Moving forward, the splenium thins out into the body of the corpus callosum. Those are your fornices going anteriorly. So now we focus our attention here. So this is the end of the fornix that's going forward. So it has to go backward and then forward once it reaches the area beneath the corpus callosum. So we are now here. Move that imaginary cleaver forward. Now we're back in the midbrain. Substantia nigra, red nucleus, that stroke that we saw earlier. Hippocampus, those are the cerebral peduncles, a connection between the cortex here and the internal capsule here, the corona radiata here, and the centrum semiovale up here. So moving forward. So the anterior part of the midbrain, there's no red nucleus here. That's the third ventricle. That's the thalamus. Keep moving forward. So stop here. So the internal capsule is behind, and then you see these two round structures at the base of the brain. So these are the mammillary bodies. So this is hypothalamus. Okay, so mammillary bodies in front of the midbrain. That's the body of the corpus callosum. That's the tail of the caudate nucleus. These are the basal ganglia, the putamen, the globus pallidus, which has two parts. The internal capsule posterior limb separates them. Separates the basal ganglia here from the thalamus here in the midline. So we move forward. Okay. Moving forward again. And now we're back at the level of the anterior commissure, connecting the two temporal lobes. If you move it back a bit, that's where you find the amygdala. If you move it back again, that's the hippocampus. So move forward again. So looking here, those are the two optic nerves at the optic chiasm. So these parts that connect with the thalamus here are your optic tracts. Moving forward. Okay. Moving forward. The caudate joins with the putamen at the nucleus accumbens. And move forward. Moving forward. Now it's just the caudate by itself. That's the frontal horn of the lateral ventricle. And then we move forward again. We're back at the triceratops. We go beyond the triceratop eyes. Now it's just the centrum semiovale. Straight gyri are down here. These are the orbital gyri. And then we're back here at the frontal poles, as you see here, and as you see here on this 3D view. All right.